I'm very happy and delighted that we can welcome an old friend and colleague, Dr. Lyle Peterson, here as a speaker today. Dr. Lyle Peterson has served as the director of the Division of Vector-Borne Diseases since 2004 at CDC. Dr. Peterson began his training in the University of California, San Diego, where he received an undergraduate degree in biology. He studied medicine at the University of California in San Francisco and completed an internal medicine internship and residency at Stanford University. He then joined Tulane University's tropical medicine research efforts in Cali, Colombia, before starting CDC's Epidemic Intelligence Service, EIS, Applied Epidemiology Training Program in 1985, which will turn out to be essential for also the RKI, as I will come to a bit later. After his EIS training at the Connecticut State Health Department, he joined the CDC's Division of HIV/AIDS, where he worked until 1995. That's also when we first met, I think, at the AIDS conference in Florence, Italy. During that time, he also completed CDC's preventive medicine residency program, received an MPH degree from Emory University, and served in several posts, including as the chief of the HIV seroepidemiology branch. From 1996 to February 2000, Dr. Peterson guided Germany's efforts in creating a new national infectious disease epidemiology program at the Robert Koch Institute. So this I will elaborate a little bit because that was also after a major crisis, the HIV/AIDS crisis. There was no infectious disease epidemiology in Germany. There was a concerted action in the government that decided infectious disease epidemiology needs a booster in Germany. And so we got support from US CDC to build an infectious disease epidemiology here in Germany. And I can say that without Lyle, I think I wouldn't be standing here, and probably we wouldn't have the Department of Infectious Disease Epidemiology as it is today here at RKI. Yes. From 2000 to 2003, he served as the Deputy Director for Science of the Division of Vector-Borne Diseases. He is author of more than 200 scientific publications. Dr. Peterson has also received several scientific awards, including the Charles B. Shepard Science Award, the Alexander D. Langmuir Award, James H. Nakano Citation, the Larry J. Anderson Award for Outstanding Public Health Services, and twice the HHS Secretary's Award for Distinguished Service. He served on the National Security Staff at the White House in 2015, and in 2016 was the Incident Manager for Zika response at CDC. He is a fellow of the American Society of Tropical Medicine and Hygiene and is on its scientific program committee. He is a past president of the Society of Vector Ecology. Dr. Peterson's current research focuses on the epidemiology of arboviral and bacterial vector-borne zoonosis, which gives us the pleasure of having you as guest speaker here today. And we welcome you, and please take the floor and to give your presentation. Right.
So thank you very much, Osama, for the introduction. It makes me sound a lot more important than I really am, but anyway, it's really a pleasure to be here. And today, of course, I'll talk about climate change and vector-borne disease. So I'm going to focus this lecture on climate change and vector-borne diseases with a real focus on North America and Europe. So we'll talk about West Nile virus, which has 3 vectors in the US, Culex pipiens, Culex quinquefasciatus, and Culex tarsalis, and also about Lyme disease in North America, which of course the vector is Ixodes scapularis ticks. We'll also focus on the similar diseases in Europe, West Nile, which is vectored by Culex pipiens, Lyme disease, which is vectored by a related tick, Ixodes ricinus, and Tick-Borne Encephalitis, which is vectored by Ixodes ricinus and Ixodes persulcatus. So in today's lecture, I'm going to try and answer 4 questions. How does weather impact the ability of mosquitoes to transmit West Nile virus? And I'm focusing on weather and mosquitoes because weather is much more sensitive overall to mosquitoes than ticks because of the shortened life cycle. Then I'll talk about what is the impact of weather on West Nile incidence and trends, what we know so far. And then what is the evidence that climate change has impacted vector-borne disease incidence in North America and Europe? And ending, what is the projected impact of climate change on vector-borne disease incidence? So just to get a little bit of background, this is the predicted increase in global surface air temperature by what's called SSPs, or shared socioeconomic pathways. And these SSPs are basically estimates of how much greenhouse gas emissions the Earth will produce in the upcoming years. And so it's rated from SSP 1 through 9, which is basically the most optimistic scenario, to SSP 8.5, which is kind of an extreme amount of greenhouse gas emissions. And so you can see on this chart that depending on what scenario you want to believe is going to happen, the temperature will rise up to about 3.5 degrees centigrade from where we are now. And so on the left axis is the temperature relative to 1995 to 2014, and on the right axis is relative to 1850 to 1900. So we've already increased the temperature since then about 1.3 degrees centigrade. And so the point I want to make with this is that changing the temperature even 1.3 degrees centigrade, certainly by 4 degrees centigrade, is going to have an enormous impact of vector-borne disease. And so I'm going to talk a little bit at first about how that is going to impact what we know about mosquito biology. So along with the increase in temperature there's going to be a predicted change in mean precipitation change relative to 1850 to 1900. And on these 3 graphs, I've indicated the percentage of change that's been predicted, either 1.5 degrees centigrade global warming, 2 degrees, and 4 degrees. And what you can see from this chart is, I think I can show this, is that in northern North America and in Europe, under all of these scenarios, that rainfall is going to increase. So the humidity will increase, and under the most extreme circumstances, Southern Europe will get drier. And we'll get to that later, why that is important. So I'm going to talk now about, start with what we know about the impact of temperature on what's called vector competence, or the ability of a Culex mosquito, once it's infected or exposed to the virus, to spread it. So this is the results of an experiment, and they looked at 2 different viruses. The New York 99 virus, which is a lineage 1 West Nile virus, which was the one introduced into the United States in 1999 in New York City. The other virus is West Nile virus 02, which took over starting in 2002. So the original virus of New York 99 disappeared, and now it's descendants of this West Nile 02 virus that now circulates in North America. So the way this experiment was done was they took some Culex mosquitoes, they orally fed the mosquitoes West Nile virus, and then incubated those mosquitoes at different temperatures ranging from 12 degrees centigrade to 30 degrees centigrade. And after 5 days or 10 days or 15 days, allowed those mosquitoes to feed on mice. And so the experiment was, how often do those mosquitoes transmit the infection to mice? In other words, known as vector competence. So, excuse me. So if you start, let's just look at the blue lines here. So this bottom blue line is if you incubate the mosquitoes or maintain them at 12 degrees centigrade, after 5 days, none of those mosquitoes transmitted infection to mice. Whereas if you did the same experiment and incubated them at 32 degrees, about 30% transmitted to mice. And so obviously, the longer you wait, after 10 days, a higher proportion transmitted, and after 15 days, even a higher transmitted. So what you can see from this slide is two things. One is the increase in vector competence is not linear. So if you increase the temperature by 1 degree centigrade, you get an exponential increase in infectivity of the mosquitoes. So in other words, a small change in temperature causes a big change in the ability of the mosquito to transmit infection. So the other thing to take away from this is this New York 99 differs from the West Nile 02 in that, so for example, mosquitoes that are kept at 30 degrees centigrade, they are less likely to spread West Nile virus 99 than West Nile 02. So the West Nile 02 virus has a higher vector competence at the same temperatures. And that's why the virus actually took over. So point is that small increases in temperature cause big differences in the way these mosquitoes can transmit infection. So the same experiment was done with European mosquitoes and European virus, which is a lineage 2 virus mainly circulating in Europe. So in this experiment, it was slightly different in that they either injected the mosquito with virus or allowed them to orally feed. And then they saw how often they saw the virus in the mosquito saliva. So in this top line up here is if you inject the mosquito with virus, 100% of them are infected, no matter what temperature. And if you inject them with virus, they all transmit infection, no matter what the temperature is. But if you orally feed the mosquito virus, what you see is as you increase the temperature, generally the more of the mosquitoes actually become infected and more of them transmit. So basically the same finding. The higher the temperature, the higher the vector competence or the ability of the mosquito to transmit. And it doesn't take a huge temperature change to do that. Okay, oops. But vector competence isn't the only factor in here. It's really what we call the vectorial capacity, which is the ability of a mosquito to pick up the virus and transmit it in nature. And so, of course, with the West Nile virus transmission cycle, the mosquitoes emerge and the female mosquitoes go into at the end of the season, go into a phase called diapause, which is basically hibernation phase. And those female mosquitoes are infected. So if you go to a place where West Nile is circulating, in the middle of winter, it may be freezing cold. If you go into the sewers of the city or find a warm place, you will see mosquitoes resting on the walls of sewers in any warm place. And some of those female mosquitoes are actually got infected the previous summer. And so when the temperature warms up, those female mosquitoes emerge. They bite birds, which are the primary hosts. Birds get infected, more mosquitoes feed on those birds and get infected, and this cycle continues round and round over the course of the summer, and usually peaks about the middle of August, where you have the most infected mosquitoes. And humans and horses are dead-end hosts and don't have anything to do with this. So factors that influence this transmission cycle increase the ability of West Nile to cause outbreaks. So this cycle in reality, although on the last picture seemed very simple, is actually really complicated. And it's affected in multiple ways by temperature and rainfall. And so I'll go through this complicated slide. So first of all, the more mosquitoes you have, the bigger problem you have. But rainfall does two things. One is it increases the breeding sites, or the places where the mosquitoes, immature mosquitoes, can breed. But it also has a negative effect. So if you have too much rain, it just washes out the system. And so rain, depending on how much you have, either can promote mosquitoes or actually decrease them. So for example, during a hurricane or a big rainfall event, we actually see no increase in vector-borne disease because it's basically flushed out all the breeding sites of mosquitoes. Winter freezes can cause the mosquitoes that are in this diapause phase to die, so that has a negative effect. Temperature does two things in opposite directions. One thing is it increases the development rate of mosquitoes, so you get basically more mosquitoes the higher the temperature. But on the other hand, if it gets too hot, it increases mosquito mortality. So it also has a differential effect. If you have droughts, in other words, a big lack of rainfall, it does an interesting thing. One thing is it can reduce the predators that are feeding off the mosquito. So it increases the mosquito abundance. But at the same time, it decreases the breeding site, so it decreases mosquitoes. But it also does another thing, is if you have fewer pools of water in an area, that's where all the birds go, and then the mosquitoes are also there. So it congregates or puts together the mosquitoes and birds, which are infected, and that can increase the infection rate in the mosquito. Also, temperature increases the biting rate of mosquitoes, so the mosquitoes bite more, so they have more of a chance to get infected. But they also increase or decrease this, what's called the extrinsic incubation period, or the time it takes from a mosquito to become infected to the time it becomes infectious. So temperature affects that. And all of this increases the amount of virus in mosquitoes. So if you have a combination of a lot of mosquitoes and a higher infection rate in mosquitoes, you have a big problem, which then relates to human cases. And of course, if a lot of humans are infected, you only get West Nile once. So you have less human cases if a large proportion of the population is actually immune. So in essence, the takeaway from this is really complicated, and the effect of temperature and rainfall have differential effects depending on how much temperature increase there is or how much rainfall or increase or decrease there is. This makes it very hard to predict what's actually going to happen. So let's talk about this concept I mentioned about extrinsic incubation period. So a mosquito is not a syringe. So once a mosquito picks up the virus has to replicate in the midgut of the mosquito, go into the hemolymph of the mosquito, which is the equivalent of the bloodstream of the mosquito, go to the salivary gland, and then have virus in the saliva. Well, that takes time, and it's very influenced by temperature. So going back to the West Nile transmission cycle is the female mosquito emerges as an adult, she mates, and then she feeds on an infected bird. So the mosquito at that point picks up the infection. She then lays her eggs and then goes for another blood meal. And once a mosquito is infected, it is infected for life. But if the extrinsic incubation period, or the time it takes for that mosquito to get infected, is a long time, when it takes the second blood meal, it's not infectious. So no transmission happens. Also, the mosquito has to live this long to take a second to bite to bite twice. So there's no possibility of transmitting an infection if the mosquito doesn't live this long. Now, on the other hand, if the temperature is warmer and that extrinsic incubation period is shorter, what happens is, again, the female emerges an adult, she mates, feeds on an infected bird, gets infected, lays her eggs, but the next time she feeds on a person, she's now infectious. So she transmits the infection at that point. And she also doesn't have to live very long to be able to take that second blood meal. So this extrinsic incubation period is really, really critical for West Nile transmission in nature, and it's temperature dependent. So the best way to lose an audience is to present a complicated equation, which I'm now doing, so please stay with me. So this equation describes vector competence, or effectively what we can kind of think of as the R0 of the mosquito world. And so it's really a measure of the number of infectious bites delivered to a population at risk. So it's in this complicated formula, which determines vectorial capacity. But all the parts of this equation are all influenced by temperature. So this part of the equation, temperature affects the vector abundance, it affects the host contact frequency. It also affects the impact of mosquito survival among infection, and also impacts the extrinsic incubation period, which we just talked about. But notice in this equation, this extrinsic incubation period is a logarithmic function. So the shorter that period, the infection rates in the population go up logarithmically. It also affects the vector competence, as we talked about earlier. So temperature affects all of these things. So what can we conclude from all of this? One is temperature increases the vector competence, or the ability of a mosquito to transmit infection. It's not a linear increase, so a small increase in temperature produces a big increase in vector competence. Temperature also has many other impacts on West Nile transmission, which I explained in that complicated diagram. It shortens the extrinsic incubation period, so mosquitoes become more infectious faster, but it also increases the viral replication in mosquitoes, so you have more virus in the mosquito, and it also impacts mosquito survival, mosquito replication and development, and the interactions with hosts. So it's all very complicated. So what do we know about what's happening with West Nile incidence and distribution in the United States? So this is what's happened since West Nile virus was introduced in 1999 in New York City and then spread across the country. So the first 3 years, not too much happened. There was very few cases of West Nile virus. So also think of Germany, which also has West Nile introduced in the last few years. Just because nothing big has happened doesn't mean it won't happen in the future. As the virus spread across the country, it had some huge outbreaks, and this is the number of neuroinvasive disease cases only, and has gone up and down ever since with no clear pattern. But what I can say is in 2002, we had a heat wave. In 2012, we also had another heat wave. So temperature seems to have an effect on what happens. So this is the pattern. So let's start with the graph on the left. This is the pattern of West Nile virus in the US so far. And this is by county, and we have about 3,000 counties. And you can see that West Nile virus is really prevalent in certain parts of the country and not others. But if you also compare it with the temperature, this is the summertime temperature in the US, and this is the annual rainfall in the US. And so you can see that there's not a clear correlation between temperature and rainfall of where West Nile virus occurs. So obviously there's some other ecological factors that are involved in determining where West Nile is occurring, and we really don't fully understand why this pattern occurs the way it does. So I also want to mention this part of the country, while it's reasonably warm in the summer, is also the coldest part of the country in the winter. So you can have very cold areas that still have a lot of West Nile transmission. I mean, cold being in the winter. So what somebody did is they took that long equation I showed to you earlier and then tried to model all of the factors that are involved in that equation. And what they did was they came across, they predicted that West Nile virus, using, depending on which mosquito species, which Culex mosquito species, would peak around 25, with a summer temperature around 25 degrees. And lesser summertime temperatures would not promote West Nile transmission, and higher temperatures would not either. So they predicted that West Nile virus would peak around 25 degrees by modeling that long equation. And so this is actually what happened. So this is, let's start with this line here. So this is the line of West Nile virus incidence for per 1,000 people for 3,109 counties in the US. And so this is mean incidence, and this is the average summertime temperature. And if you follow this gray line, basically what they found is the counties with the highest overall West Nile incidence over a 16-year period, in fact, had a mean summertime temperature of about 25 degrees. On this graph, they've also, just for illustration, put 42 counties with standard deviation around how much West Nile is actually occurring. And you can see that even though there's some tremendous variation from year to year among many of these counties. But the point is overall, if you take the whole country, it looks like the transmission maximum is around 25 degrees centigrade in the summer. So what a number of other people have done is they've said, okay, so maybe 25 degrees is optimum overall, but what happens from year to year? How do you explain West Nile incidence from year to year? And so 4 people have looked at West Nile virus in the US, mainly in the northern parts of the US, and saw what variables correlated with more West Nile cases. And what they found was that in all of these cases, a higher summertime temperature correlated with West Nile outbreaks, with a variable effect on humidity and rainfall. So within this endemic area, overall 25 degrees centigrade seems about maximum for overall West Nile transmission. But within those areas, if you have higher temperatures, you get more West Nile. So heat waves promote West Nile outbreaks. So another group of people looked at, well, what other variables seem to influence West Nile transmission? So what they did was they took all the US states over a 10-year period, and they saw what variables seem to correlate with more West Nile. Population immunity had an effect, of course, because people are immune, they don't get West Nile. Rainfall actually decreased slightly overall, the amount of West Nile. So drier conditions, if you take the whole country, seem to have a negative effect. And if you looked at drought, where you had extreme decreases in rainfall, that was actually the best predictor overall of how much West Nile virus you had. And temperature, as we talked about earlier, temperature overall, the hotter it got, generally, there was more West Nile. So in 2011, we had the world's largest West Nile outbreak, which actually nobody paid attention to because everybody was worried about, or 2021, everybody was worried about COVID at the time. But this was a huge outbreak. There were 1,600 reported cases. We had almost 1,100 neuroinvasive disease cases, 113 deaths. And the outbreak occurred in Phoenix, Arizona, which I've pictured here on this slide. And so you can see that Phoenix, Arizona is a very dry, hot place. And so I've taken this map of West Nile temperature in the summer, and where this arrow is Phoenix. So it is like one of the hottest cities in the US. There's about 3 million people that live there, and West Nile is crazy rampant in Arizona. But during this outbreak, with about 1,000 neuroinvasive disease cases, we estimate that about 5% of the entire population got infected. So think about it, 5% of the entire population of millions of people all got infected in about a 4-week period. I mean, it's really amazing. So what happened to cause this outbreak? So this is the temperature of Phoenix over a 20-year period. And this dark red line is what happened in 2021. And correspondingly, here's the temperature anomalies for the same years. And you can see that it was really hot. It was like 35 degrees in the summer, but it wasn't abnormal. But what was abnormal was here, which is rainfall, and this is in millimeters. And so you can see that during the summer months, that there was in June or July, August, and September, there was an abnormal amount of rain, and as you can see from this anomaly. So even a small amount of rain in this desert area under a very high temperature caused a massive outbreak. So going back to this complicated slide, people have also tried to model this. So what they've tried to do is take all of these factors and figure out, well, what do we know about each one, and then make a model of each of these things and combine it and try and figure out how much West Nile we could actually predict. So they did this up to about 2014. And so ignore this. This is the death rate, but ignore that. So this is the number of cases per year for the US up until 2014. And then they took, using that complicated diagram that I just showed you, tried to model the predictors of what would happen if temperature increased. And so what they said was, okay, the mean current number of neuroinvasive disease cases up until that point was about 1,300 a year. 95% of the time it was up to about 2,000 a year. And what they predicted using 10 climate-based models was what would happen in 2036 to 2049. And what they predicted was that the number of cases would increase about 75%. And under an extreme scenario, even higher. So just a model suggested, well, okay, in the next 20 years, given the expected temperature increase and rainfall changes, that we would see about a 70% increase in West Nile cases. So what can we conclude from all of this? So the West Nile distribution is determined by a complicated set of ecological factors, of which climate is just one. On a continental scale, areas with summer temperatures around 25 degrees are associated with higher incidence, in endemic areas, increased temperatures generally are associated with higher incidence. And the relationship with rainfall is complex, as what I mentioned. Overall, drought or decreased rainfall is associated with higher incidence. However, in dry areas, increased rainfall has produced massive outbreaks. So if it's dry, more water is good. If it's wet, more water is bad, is basically it. And so modeling also suggests that West Nile incidence may double or close to double in the next 20 years. Okay, so what's happening? What do we know about the influence of climate on West Nile virus in Europe? And as you'll see, it's a very shockingly similar kind of scenario. This is what the incidence of West Nile cases by year in the European Union from 2008 to 2011. And I know you're all familiar with this great big outbreak in 2018. But generally, if you look at this graph, sort of suggests that West Nile virus is generally going up. So I think one of the shocking things to me is how much the temperature has actually increased in Europe. And this is European annual surface air temperature anomaly. So compared to a period of 1991 to 2020, this is the difference in overall temperature per year. And what you can see is back here, you know, relative to this reference period, temperatures were about 1 degree to 1.5 degrees centigrade lower. But all of a sudden in recent years, you have this massive increase. So really from comparing back here to over here on the right, there's actually been about a 2 degree centigrade increase in overall temperature, which is huge. And this is actually what you would predict, because so far we know from global warming about 1.2 or so degrees temperature has increased worldwide, but it's expected to increase disproportionately in more northern and southern areas. And so if this was pretty scary to me when I started looking at this data. Now, this is the same kind of a graph, except this is just looking at summertime temperatures through 2018. And what you can see is summertime temperatures have increased even more dramatically. And then 2018 was the warmest summer on record, which corresponded with this massive outbreak. So again, like in the US, it looks like more temperature produces more West Nile. And what was also striking was that the temperature in 2018 was a year-long event. So this is the seasonal maximal temperature anomaly in Europe from winter, spring, summer, and autumn. And you can see that no matter where you were in Europe, throughout the entire year, it was exceptionally hot. But what happened with rainfall was somewhat different. And so in that year, and we'll just look at summer for now, what you can see is that and down here in Greece and Macedonia, it was really rainy. And in fact, they published a number of papers from that area saying, see, our big West Nile outbreak was due to this really hot temperature combined with this rainy period. But in more northern parts of Europe, it was completely different. It was very dry. So, and that also promoted outbreaks. So again, going with this idea that if a dry area like down here, if it gets more rain, that promotes West Nile, and a more moist area like Northern Europe, less rain actually promotes West Nile. So rain has a differential effect, just like it does in the US, does the same thing in Europe. And so what happened as a result of all this is West Nile virus cases beginning since the beginning in Europe. And it's no surprise to you that there is more West Nile suddenly started emerging in 2019 in northern Germany and in the Netherlands, and it's persisted. And so, but actually during that 2018 outbreak, enzootic activity, or activities in birds and mosquitoes, actually was known to start about that year. So that preceded the human cases by a year. So all of a sudden, because of these increased temperatures, you all of a sudden saw this very marked northern spread. Now, there's not very many cases in Northern Europe yet, but keep in mind that when West Nile came to the US, first 3 years, we hardly had any cases either. So I don't think, we certainly don't know what's going to happen in the future, but this dramatic increase certainly seems related to these increasing temperatures that I've described. So another person looked at all of Europe from 2007 to 2019, and then took a model and tried to look at all the temperature and West Nile data and tried to predict what would impact West Nile in Europe, or what has impacted West Nile in Europe. And what they found was West Nile seemed to be associated with places that had a mean winter temperature between 2 and 6 degrees. And remember that the female infected mosquitoes, the virus lives in female infected mosquitoes during the winter, and they try and find some nice warm place. And it turns out the optimum warm place is between 2 and 6 degrees, so this makes some sense. If it's too cold, they freeze. If it's too warm, they don't do well either in this hibernating state. They also found that higher temperatures, meaning an average summer temperature above 22 degrees, was related to more West Nile in Europe. Now remember, in the US, they modeled it and it looked like it was about 25 degrees. So it's very similar. If you've got a temperature somewhere 22, 25 degrees, that seems to be about optimal. But what they also found is places with higher incidence had more rain. But on the other hand, places with higher incidence had more or less surface water extent. In other words, less standing water on the ground, which also goes along with the fact that if you're dry, more rain promotes West Nile, and if you're wet, you don't want more rain. So very similar kinds of findings that you find in North America. So what conclusions can we make? Europe has experienced increased temperatures in recent years, very dramatically, particularly in the summer. The large outbreak in 2018 occurred during an exceptionally warm year, and the association with rainfall was mixed. Some areas experienced increased rainfall and others had decreased rainfall. And on a continental scale, incidents associated with areas with summer temperatures greater than 22 degrees centigrade overall, more rainy days in the summer, but with a lower surface water extent, and mean winter temperatures between 2 and 6 degrees. And enzootic activity, meaning circulation of the virus in birds and mosquitoes, as well as human cases, spread markedly northward in conjunction with these increased temperatures. So now I'll switch over and talk about Lyme disease and move over to tick-borne disease. So this is the life cycle of Ixodes scapularis, and the life cycle of Ixodes ricinus in Europe is very similar to this. And so it's a multi-year, 2-year cycle. The eggs emerge in the spring. They molt into larvae, feed on small animals. They overwinter, emerge as nymphs at year 1. These nymphs are what causes most of the infection to humans, because the larvae pick up the infection here. So these nymphs are already infected. They molt after taking blood meal, turn into adults, mate on deer, and feed on deer, and then she lays her eggs, and the whole cycle starts over again. So it's a 2-year cycle, but because it's a 2-year cycle, it's not as sensitive as to acute changes in temperature like what you get with mosquito-borne diseases. But it's also a lot easier to kind of figure out what's happening with overall changes in climate. So there's a lot of impact of climate and weather on Lyme disease. First, the minimum temperature primarily defines the northern distribution of vectors. So the warmer it is, the more north these Ixodes species can go. Warmer temperatures increase the reproductive capacity of the ticks. In other words, they can reproduce better. leading to larger population and a greater risk for human transmission. They're very sensitive to moisture. So these ticks require about 85% humidity. Otherwise, they dry out and die. And so the more moist it is, it allows for tick survival in warmer environments. Temperature and moisture also affect the tick behavior in different ways. And I'll talk about that in a second. And temperature, meaning the number of days it's warm enough for the tick to be active, affects the disease seasonality. So the longer it's warm, the more likelihood it is that those ticks can bite people. So the way these ticks actually feed is they hang out or they stay down in the leaf litter in the forest where it's kind of moist and near the ground. And then what they do is they climb up blades of grass or bushes and just hang out at the top. And that's called questing. And so what they do is they sit up here and wait for some animal to come by, and they can detect the temperature of the animal. And when the animal comes by, they jump off, land on the animal, and then feed, or land on you and feed. So, but this questing behavior is complicated for a tick. One is it wants to feed, so it's got to go up here and quest. But when it goes up there and quest, the humidity is a lot lower. And so there's a danger that that tick is going to dry out while questing. So it's kind of a balance. Increased temperature causes the ticks to want to quest. But on the other hand, they have a greater likelihood of drying out. So temperature has a very complicated relationship with these ticks and their survival. So this is what's happened with the change in temperature in the US and relative to 1901 to 1960. And I want you to pay attention to two areas, which we'll talk about in a minute. Lyme disease is found this area and in this area. So you can see where Lyme disease or these ticks, Ixodes scapularis ticks, are present. It's gotten warmer. But in the same areas, you can see that it's also gotten wetter. So in these more northern areas, it's gotten warmer and wetter, which is a good thing for ticks. And as a result, what's happened is that the Lyme disease incidence, which is shown in green, this is the incidence of all tick-borne diseases that are reported to us, is going up and up and up. So we've had this huge increase in Lyme disease cases. And we now have about 36,000 cases a year reported to us. But we know from other data sources that the true number is about 500,000. So there's actually about 500,000 Lyme disease cases in the US and increasing every year. And what's happened is on this map is Lyme disease cases. Each dot represents a case in 2001 and 2019. And what you can see is this dramatic geographic spread of Lyme disease. So which started out about here has now gone north into the more northern parts of the country as it's gotten warmer and wetter. But also Lyme disease has also gone south too. So climate is not the whole story. I can't explain how climate would make Lyme disease go south. But clearly the conditions in the northern part of the country have become more favorable to ticks. And so this is what we know about the distribution of Ixodes scapularis, comparing 1996 to 2000. And what you can see here is this dramatic increase in the northern spread of the tick, corresponding to increased temperature and increased rainfall. You can also see that there's a lot of Ixodes scapularis down here, but there's no cases. And that's because actually these ticks have a different, although it's the same species, don't bite humans. And that's another long story. Now, what also people have done is they've tried to look at, this is a study that we did, looked at the possible effects of climate change on when these ticks emerge in the spring. And so if you model this out and look at what you might expect over the next 20, 30 years, here's what's baseline. These are under different climate scenarios. RCP 8.5 is high greenhouse emissions, 2.6 is less. But what you can see is the more greenhouse emissions, the more temperature increase, that the week that these ticks emerge and Lyme disease starts happening goes down, becomes earlier in the year. So as the temperature goes up, you have a longer season where ticks are active and potentially can bite people. So what can we conclude from all this? In North America, the incidence of Lyme disease is increasing. It's going northward, but also southward for other reasons. The northward spread is associated with the northward distribution of the tick vector. Areas where the tick is spreading are getting warmer and wetter, making it conducive for the survival of the ticks. And climate change is expected to increase the length of the tick season, putting more people at risk. So let's see what's happening in Lyme disease and tick-borne encephalitis in Europe. Oops, keep pushing. So this is the known distribution of Ixodes ricinus in Europe. As you can see, it covers much of the continent, going all the way fairly north. Now, so what people have done is they've tried to see what's happened with the distribution of this tick over a long period of time. And I think this is the best study around. It's actually in Russia, but I think it is indicative of what's happening in the rest of Europe. And what these people did was unbelievable. They must have had good grant funding or something, but they studied the Ixodes ricinus period over a 35-year period in two areas. One was a deciduous forest, and one was a broadleaf forest. And what they did was they looked at the ticks per 1 kilometer of transect from 1975 to 2015 or so. And what you can see is the number of ticks in both of these regions has increased dramatically over time, over this 35-year period. And if you look at the temperature of these two areas, you can see that overall, the number of degree days, which is simply the temperature times the number of days that it's at that temperature, has gone up over time. And so in this area, it's gotten warmer. And what they also did was they correlated how warm it was, or the number of degree days in the summer, with the number of ticks they found. And what they found was in these two areas, that as the number of degree days goes up, the number of ticks also goes up. So it was a really good long-term study showing basically it's gotten warmer, and as it gets warmer, the number of ticks actually goes up. So in another study in the Czech Republic, which was basically the same idea, except they were looking at in the mountains, and what they found was overall in their study area that the mean temperature went up over time, a temperature of 1,000 meters, about 1.5 degrees or 2 degrees. And over the time, what they found was the maximum altitude of finding Ixodes ricinus ticks went from about 700 to 800 meters to about 1,100 meters. So as the temperature is warming, these ticks are actually moving north to a higher elevation. And so on a larger scale, looking at Sweden, what they did in this study was they put out a survey of residents and asked people to report ticks, basically. And they did this survey twice. Once was in the 1990s and once was in 2008. And so here's the distribution of where people reported ticks in 1990s and in 2008. And you can see that, you know, the approximate upper limit was around 60 degrees north, but it's gone much further north. And this data has actually been corroborated by people actually going out and purposefully looking for ticks. So in Europe, Ixodes ricinus is moving north as the temperature is getting warmer. And so what people have tried to do then is, well, okay, if the temperature is getting warmer, how can we predict the expansion of Ixodes ricinus? And so on these slides, look at these purple areas. This is the present situation where Ixodes ricinus is, and looked at what would happen in 2050 or 2070, depending on what climate extreme you wanted to take. But overall, what they said was, looking at these, and I know it's a little hard to see on this graph, was that the range expansion was about 10 to 11% under these two climate scenarios. And what they predicted was in the Alps right now, you can see it right in there, there's not a lot of ticks because it's higher elevation. And as the temperature warms up, those ticks, the Alps becomes a risky area. And some of these areas in the south become no longer so risky, and areas in the north become more risky. So it's kind of what we'd predict and what's already been happening. So what's happening with this expansion of ticks, particularly northward, is reflected in Lyme disease incidence and trends. And it's very difficult to kind of actually figure out what's happening with Lyme disease in Europe because surveillance systems vary from country to country, and there's not a lot of long-term data that's reliable. Nevertheless, if you look at all the papers that have been published about Lyme disease incidence and combine it with surveillance reports, you end up with a graph that looks like this. And what you can see is that these arrows point to the yearly change in incidence. As predicted, the biggest change in incidence of Lyme disease is in the more northern areas where the tick is expanding its distribution. Tick-Borne encephalitis is even more complicated to look at. You see kind of a very similar kind of a picture as you do with Lyme disease, but Tick-Borne encephalitis is complicated, of course, because people get vaccinated. So, you know, I think the long-term trend data for Tick-Borne encephalitis is a little bit harder to interpret. Now, so we talked about Ixodes ricinus, but I'm going to mention Ixodes perscultatus, which is a vector of Tick-Borne encephalitis, mainly in areas like Russia, but it's been invading Finland recently. But what's happened with this is really interesting. And what's happening, of course, and so this is Sweden. And let's start over here on the right with Ixodes ricinus. Of course, the Baltic Sea is here. And so what they did was, here's areas that they detected Ixodes ricinus in this study. But there was no Ixodes ricinus up here. But looking at Ixodes perscultatus, what you now see is all of a sudden this tick has suddenly appeared in northern Sweden. And what essentially has happened is the temperatures got warmer. It allowed these ticks to go north, and now it's gone around the Baltic Sea and is now going down into Sweden. So it's pretty remarkable if you think about it. It's kind of gone through the back door in a way. So what can we conclude from all of this? Warmer temperatures are extending the period of Ixodes of activity for Ixodes ricinus and increasing the distribution in more northern areas and higher altitudes. Models suggest an approximate 10% increase in the distribution of that tick by 2050. The impacts on disease incidence are unclear due to heterogeneity of surveillance in tick-borne encephalitis vaccine, but Lyme disease incidence increase is more apparent in northern areas where you might expect. And there was a more northern distribution of Exodes persicutatus, which has allowed it to invade Sweden from the north. So what can we conclude from all of this? So to go back and take those 4 questions that I've posed in the beginning. 1, how does weather impact the ability of mosquitoes to transmit West Nile infections? So the point is even small increases in temperature increase the vector competence, or the ability of the mosquito to transmit infection. It's a logarithmic increase. It's not a linear increase. Second is increased temperature shortens that extrinsic incubation period, essentially making the mosquito more infectious faster, which has an exponential impact on vectorial capacity, or the overall transmission of West Nile in the community. And there's many other impacts on the mosquito biology, which are very difficult to figure out. Temperature and rainfall, as I mentioned earlier, has a very differential effect on all of these parameters. The second question was, what is the impact of weather on West Nile incidence and trends? And so the overall geographic distribution of West Nile is determined by a complex, poorly understood set of ecological factors. And I talked about what's happening in the US. The same thing is happening in Europe. For example, it's not entirely obvious why there isn't more West Nile virus in Spain, for example. So there's clearly other factors that we really don't fully understand that impact this geographic distribution. Nevertheless, in more temperate areas or more cooler areas within this distribution, heat waves are associated with higher incidence. Areas with an average summer temperature of around 23 degrees, 25 degrees centigrade have the highest long-term West Nile virus incidence, but many exceptions occur, and I showed you the example of Phoenix, which is a lot hotter than that. And rain increases the incidence in dry areas and decreases the incidence in wet areas. So what is the evidence that climate change has impacted vector-borne disease incidence? I think all of the data I've showed you, the laboratory data and the ecological data, suggest plausibility, meaning that even a 1 to 1.5 degree temperature increase has plausibility that it would greatly impact vector-borne disease incidence. There's northern spread of the Exodes scapularis into warmer and wetter areas. For Exodes ricinus and persicutatus, there's also northward spread and spread to higher elevations in areas experiencing long-term temperature increases. Increased Lyme disease and Tick-Borne encephalitis incidence in northern areas. And West Nile virus has expanded into Europe to more northern areas coincident with increasing temperature. So what do we think is going to happen in the future? And this is where it gets more complicated. So all of the climate models suggest that more northern areas are going to get warmer and more humid, favoring expansion of tick populations. So we expect that ticks are going to continue their northward spread. Some areas will have decreases in Lyme disease incidence simply because it's gotten too hot and dry, and that would be mainly places in more southern areas of Europe. Increased northern spread of West Nile in Europe, which into Germany, as you all know. Weather, and as temperatures continue to go up, we would expect that West Nile virus, that 25-degree summertime temperature, is going to continue going north, putting more of northern Europe at risk. Weather extremes may increase the variability in West Nile virus incidence, and we talked about what happens when there's heat waves. Heat waves promote West Nile outbreaks for the most part. And there's few credible models to exist to it for the impact of climate change on vector-borne disease incidence. And so anybody that tells you, well, you know, Lyme disease or West Nile virus is going to increase 75% or 100% in the near future, there's a lot of uncertainty to all those models. First of all, as we talked about, there's many parameters that impact vector-borne disease incidence, and many are impacted differentially by weather and climate, and many are hard to measure. Like, it's really hard to go out and measure bird immunity, for example. And no model has taken into account what happens with climate change on host populations. Like, no model has figured out what is going to happen to bird populations, for example, if the temperature goes up 2 degrees, or what happens to mouse populations in the case of Lyme disease if the temperature goes up. And so no model has really accounted for that interaction between the virus and the principal hosts. And further improvements, and this is an advertisement, and what I want to say is that further improvements in environmental data collection and disease and climate modeling efforts applicable to future climate scenarios should be a priority. We really don't, we need better models, we need better ecological data to kind of sort all this out and actually figure out with any real degree of certainty what's going to happen in the future. And given what I've shown you, what's happening with climate change, which is pretty dramatic so far and going to get worse, we really need to kind of figure out what exactly is going to happen and all the impacts on the environment that's going to affect vector-borne disease transmission. I'll skip that. And so this is a notice from our lawyers saying that whatever I've told you, the CDC takes no responsibility for it. And so with that, I'll end and thank you for your attention.
Thanks, Lyle, for this lovely presentation and interesting data that you presented. So now basically you're saying, although we haven't observed a lot of West Nile so far, we can sort of count on it that it will happen one year or the other when we get a hot summer. So what would be your recommendation for local governments and also for the national government to best prepare for this?
Well, I think for one is, I think it's really important that physicians become aware of West Nile, because they may have many patients which may have similar symptoms, but nobody tests for it. And so to try and actually figure out what exactly is happening in the animal and human population is pretty important. So although horses get vaccinated, many don't. So equine surveillance is important. Human surveillance and making physicians aware is critically important, particularly for neuroinvasive disease, because all those people end up in the hospital. So you can theoretically find them all. And also, I think some preparation thought about what kind of mosquito control or vector control you want. It's a controversial subject, I know, particularly in Europe. And so kind of figuring out, well, what are you going to do if we do have a big West Nile outbreak? Because I think it's going to happen. Sooner or later, there will be a bit, that's my guess. As I say, CDC takes no responsibility for anything I've told you, but you know, given the scenario of what's been happening and the trend, I think the possibility for a pretty reasonable West Nile outbreak happening in more northern areas of Europe is a real possibility. And thinking about what you're going to do and how you're going to respond to it ahead of time, I think is important.
All right, thank you. So do we have questions from our chat?
Or from the audience?
Or from the audience?
If this micro works?
Yeah.
Okay. Thank you, Lyle, for the excellent talk. I think very useful thoughts and data. We have the West Nile situation in Germany. It's endemic since 3 years. But the situation in Europe and North America seems to be a bit different. So in North America, it appeared that the spread was relatively swift. In the first years, you mentioned that in 4 or 5 years or so, it swept over total of the US. In Europe, Southern Europe, in the classical endemic areas, it's relatively stable. So there's not much geographic spread. In Germany so far, it has been stable as well. We don't know what will happen, but do you have any reasons, any explanations for these differences in the situation between the US and Europe?
Yeah, I don't have a really good reason. One can only speculate. Well, so I think there's a couple of things that one might consider. One is we have more mosquito species that actually spread West Nile. So you have Culex pipiens, which is similar to the northern US, but we also have Culex tarsalis, which is a really good spreader of West Nile, as well as Culex quinquefasciatus, which is also a decent spreader of West Nile. So I think there's a difference in mosquito biology that may explain some of this. Some of it might be explained by bird flyways and bird species that these mosquitoes are feeding on. But beyond that, I don't have a good explanation. Because in the US, we saw very dramatic in the beginning, north to south spread of the virus, which corresponded nicely to where birds are normally flying, north to south. But the virus's spread from east to west was a little bit hard to explain. Some bird species just fly around randomly, sometimes for hundreds of kilometers. But we don't really have a great model of how it spread from east to west. So, who knows?
Okay, so I think we have questions from our audience that's listening to us online.
Yes, thank you. This is a question from a colleague from our Department 3. Thank you for the great talk. What do you make out of the West Nile virus situation in the Netherlands, where West Nile virus was noted in mosquitoes, birds, and humans in 2020, but not at all in 2021?
Yeah, well, that's a good question. But one thing to keep in mind that 2021 was a relatively cool year in Europe. And so whether the virus disappeared and just didn't stay, or whether it just went down to a low level and just wasn't detected, nobody knows. So I think, if we have a warmer summer this summer, it'd be interesting to see what happens. Because just because you didn't detect it doesn't mean it went away. And but I do think that the cooler temperatures in 2021 actually was largely responsible for a decrease in West Nile incidence overall in Europe.
And we do have other questions in the chat too. And this would be, what are the prerequisites for human-to-human transmission of West Nile virus via vectors? And is this totally excluded not to happen?
It's pretty much excluded not to happen. Whether it could theoretically happen on a very rare occasion, I don't know. But what you see in birds is about, particularly in the species of birds that are spreading West Nile, is you see about 4 to 8 logs of virus. And what you see in humans is about 1 log or less. So humans just do not have enough virus in their bloodstream to effectively infect mosquitoes. And that's why humans, and as well as horses, are dead-end hosts. We just don't, although we get sick, we just don't have that much virus in our blood.
I have a related question to that. So what's the genetic adaptation of the virus to the humans? So may that explain the spread in the US? The better, the first 3 years, it was really low, but then it started increasing. Are there any studies to that?
So humans actually really have nothing to do with this, of how it spread, because simply, as I mentioned earlier, we just don't produce enough virus in our blood to effectively infect mosquitoes. And we've done those experiments, actually, where we fed mosquitoes on infected people, and people just don't have enough virus in them to effectively keep that transmission cycle going. So humans have nothing to do with it. But the one thing, actually going back to your question, is it could be a difference between West Nile virus lineages, which may explain some of the difference between Europe and the US. Given the fact that the West Nile virus 02 in particular, which emerges about the time that West Nile became a big problem in the US, was that, it had a higher vector competence at lower temperatures. So, or at lower temperatures. So, I think that maybe helped this big dramatic spread. It's more transmissible from mosquitoes.
Have you observed, or is there any incident to your knowledge where mosquitoes have lost the ability to spread certain viruses?
I only know that the answer to your question is in nature, no, to an extent, because I think the one difference, the one example I can give where that's the case is when people have infected Aedes aegypti mosquitoes with Wolbachia, which is an insect-borne, it's a bacteria that's present in many species of insects, not normally in mosquitoes. But if you take Wolbachia and adapt it to mosquitoes and infect those mosquitoes with Wolbachia, the mosquitoes do just fine. And it's actually spreads in the mosquito population like a sexually transmitted disease. So it spreads in the population, but those mosquitoes that are infected with Wolbachia are less likely to spread arboviruses. So it's a technique that's being used actually in various places around the world to control dengue. You infect, and we and other people are doing these trials where you're infecting mosquitoes with Wolbachia, it spreads in the mosquito population and then causes arbovirus incidence to go down. That's the only example that I can think of.
Maybe speaking of vector control, what in the ways of vector control would you suggest for the situation in Germany? And maybe also, we were talking at lunch about the situation in Phoenix. Maybe you can elaborate on that a bit.
So vector control is a controversial subject. The people have their own opinions about spraying pesticides. The one thing that we've learned is that it doesn't take a lot of pesticides to stop West Nile outbreaks if applied properly. Because if you think about it, as I explained in the West Nile transmission cycle, it starts out with a few infected mosquitoes, they infect more birds, birds get infected, which then in turn infects more mosquitoes. And that cycle continues until about mid-August, where you get more and more infected birds and more infected mosquitoes. And what we've learned is that if you spray for mosquitoes and kill that population of infected mosquitoes in August, what happens is there's not enough time for that cycle to regenerate lots of mosquitoes again. So basically one application, usually a couple of applications spread a couple days apart to make sure you get all of them, will effectively stop West Nile outbreaks. It's rather dramatic. The problem is that nobody does it until it's too late. And so when people start getting panicked about West Nile, then they start to control it, but by then 80% of the cases have already occurred. But it doesn't take a lot, and the spraying events that we've done to try and stop West Nile outbreaks, you use very, very little pesticide. And so what we've done is we've looked at human populations in these spray areas, and we can't even detect measurable levels of pesticide in people because you need very little. You also spray pesticide at night when bees and other insects that you don't want to kill aren't out. But of course, it's controversial still. People don't like pesticides, and I don't blame them. So yeah, so mosquito control works. COVID has actually complicated things a bit, because what happened in Phoenix, where they had this great big outbreak, was they were driving trucks around spraying for mosquitoes, and people thought they were spraying COVID and trying to kill the population. So people were throwing rocks at the spray trucks. It was kind of a bad situation. So, obviously public opinion about any kind of public health intervention is evolving in this era of COVID.
So here we have some more questions from the chat regarding the host populations. One big issue at the moment is the loss of biodiversity. So theoretically, vector-borne diseases will increase. However, in the case of West Nile virus, hosts may disappear or their numbers may decrease. Birds because of the loss of insects. Has this been taken into account in the models?
Yeah, and the answer is no. Because when I mentioned, all these models are really based on fairly static assumptions. And one of the static assumptions is that the underlying host populations, like mice in the case of Lyme disease, or deer, or bird populations in the case of West Nile, nobody has taken into account in any of these models potential changes in those host populations as a result of climate change. And I think that's a real limitation for us to try and figure out what's going to happen in the future. So I think looking at, the effects of climate change on host populations, which nobody's really done, is something that really we need to start thinking about. It's a great question.
And maybe I can continue with a question from the chat. Thank you for this very interesting talk. Do you think that transborder transportation of animals is also contributing to the introduction of ticks into new areas?
Potentially, yes. But I think the more important spreaders of ticks, first of all, ticks don't move very far. They basically stay in the same place by themselves. And so the things that are really spreading ticks around are probably other animals. So birds are infected by ticks, and birds can fly a long way. So birds are probably very good tick spreaders across long distances. As I showed with the Lyme disease transmission cycle, that deer, the adults mate on deer, and deer walk around. And sometimes for some good distance. So I think deer are also pretty good spreaders of the ticks as well. But for long distance travel, probably birds.
Regarding Lyme disease, you showed that it spreads to new geographic areas and incidence increased. Are there any specific information campaigns also, or do you do anything in regard to prevention, or do you just rely on the media that they inform the public?
So that's a really good question. It's actually just broader than where Lyme disease is going. It's where it's been, because where it's been, it's still there. It's not going down at all. And of course, we're trying to educate people about ticks moving into their areas, so they watch out for ticks. But we've put a lot of energy into figuring out how to control ticks, and it's been a total failure so far. We just don't know how to do it. There's a lot of ticks out there, and so we've tried a number of interventions. None of them. Some of them may work on a small scale, but none of them are scalable. And so tick control has been a real problem. So what we've done is tried to put a lot of emphasis on educating people to do tick checks after they've been outdoors, because it takes, roughly 48 hours of a tick being on you to spread Lyme disease. So there's a real opportunity for people to take a shower after they've been outside and pull off all the ticks. Obviously, some people will do that and other people won't. But really it's kind of making people aware and trying to look for ticks and doing tick checks that we're really emphasizing, because we just don't know how to yet control tick populations on a scalable way.
I think I have one more question here from that was given to me by colleagues. If you can say something about the status of CDC's dengue vaccine development.
Right. So right now there's in the US anyway, there's a licensed dengue vaccine made by Sanofi, which for those of you who are familiar with the dengue world, it's got its problems, because if you vaccinate of people that have never seen dengue before, and then somebody gets infected again, it causes immune enhancement, causes people to get worse dengue. So you can really only vaccinate people that have already had dengue once, which is obvious limitation. So that's the vaccine that's out there. So there's a CDC vaccine that's now being manufactured by Takeda. That vaccine is finishing its phase 3 clinical trials, and we expect, and that vaccine does not appear to have this immune enhancement problem. So that's a good thing. And it's also going to be licensed for a much broader age population. The Sanofi vaccine is only licensed for children. The Sanofi or the Takeda vaccine or the CDC vaccine will basically be available for much broad adults as well as children. And so we expect that vaccine to be available probably in the next 1.5 to 2 years. So that's a good thing. If it continues to look good in terms of not causing immune enhancement, that vaccine will probably be the major vaccine, at least for the short run.
All right, thanks. I think we have more questions.
Yeah, we have 2 more questions from the chat, a little bit more general, but maybe I can address them to you also. Besides West Nile virus and Lyme, which are the diseases you worry most about concerning population-wide impacts from climate change in the United States? And the other one would be, what factors drives West Nile virus neuroinvasive disease in dead-end hosts?
What factors drive West Nile in invertebrate hosts? What factors cause neuroinvasive disease in humans, for example? So that's another great question. And it really involves of how does West Nile virus get into the central nervous system? And there's at least 3 ways that I know of that people have hypothesized of how that happens, of how the virus gets beyond the blood-brain barrier and it affects the central nervous system. So there's a number of mechanisms that have been postulated. Which one is correct, I don't know. Certainly the virus is really good at evading the interferon immune response, and that's a very principal way of it getting past or causing neuroinvasive disease in humans. So it's a complicated thing. I'm not an immunologist, don't claim to be, but there's probably a number of mechanisms that allows that virus to get into the central nervous system. But all of these flaviviruses are really good at infecting people because they all can interfere with the human interferon response. And that's a very significant way they become pathogenic. What was the other question?
Which are the diseases you worry most about concerning population-wide impacts from climate change in the US?
Yeah, that's a great question, and I don't have a great answer because I mean, who would have predicted West Nile virus to show up in New York City? I do worry about Usutu virus. It's not going to kill everybody on the block, but it'll freak people out when it shows up in the US, and it will eventually show up in North America. I'm sure of it eventually. There's Myerovirus, which we are concerned about. I'm not too concerned about dengue. There's a lot of written about dengue going north, but I don't think that's going to happen. Dengue may go. The climate models all predict that dengue is going to go northward, but what's happening with dengue is really interesting, because dengue, which is a human mosquito human transmission cycle, what happened? But it bites. That Aedes aegypti mosquito bites people indoors, and so what happens is that along the U.S.-Mexico border, we see these huge dengue outbreaks on the Mexican side of the border, but very few number of cases on the U.S. side of the border. So all you have is a little river between the two and a border wall, but much more dengue on the Mexican side of the border and a lot less on the U.S. side of the border. Well, it actually turns out there's more mosquitoes on the U.S. side of the border than there is on the Mexican side of the border. So why not more dengue? And the reason is because everybody in the southern U.S. in these really hot areas either has screens on their windows or uses air conditioning. So their windows essentially aren't open, so there's no mosquitoes flying in their house. And people in the U.S. live further apart on the average than people in Mexico. So although climate models would predict that dengue is going to go into more northern areas, there's other human factors which prevent it from doing so. It's the same thing with malaria in Europe. Every climate model would say, oh, malaria is going to invade southern Europe. Well, malaria used to be in southern Europe, and they got rid of it. And it's just basically, the healthcare system and the way people live is preventing it from taking off in southern Europe again. So, it just points to kind of a limitation of just looking at models and predicting what's going to happen with vector distributions based on temperature, because there's other human and other environmental drivers of these things that really can affect them.
Okay, and thank you very much, Lyle. And I think with this outlook, we will close this session for today. Thanks again for coming here and taking the stress for traveling after the long time of probably very little travel. And we've been very happy to welcome you here as a guest speaker. Thank you again for your very informative talk and the discussions we had. And I'd like to encourage all those listening online to come for our next talk in person, because now we will go for refreshments.
Yeah,
Bye-bye.
Thank you to all of you brave souls who have come here in person. We have the same kind of situation here with our hybrid system. It's a lot easier to watch things at home than coming into an audience. But so thank you for coming in person, and thank you for those of you who are online.